Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A paper from 2020. So this is paper 1. Which of the following statements, A to D, about differential staining is not true? So differential staining can distinguish between different organelles. So which of the following statements, A to D, about differential staining is not true? So differential staining can be used to distinguish between two different types of organisms that are hard to differentiate. So for example, like different types of bacteria, like gram staining. But as well as that, it can be used to differentiate between different organelles as well. So if we look at A, differential staining can distinguish between different organelles. That is true, so it can't be this one. Differential staining can be distinguished between different types of cell. That is true because that's one of its uses. Differential staining can distinguish between different types of organism. That again is true. So differential staining is a common feature of electron microscopy, must be the untrue one. And also, to be honest, in most cases it's using a color thing, and you can't use color in electron microscopy, so it's D. Next, so it says which of the following statements, A to D, about microscopes are correct? So it says a light microscope with an eyepiece lens of magnification 10 with an objective lens of times 50 will have an overall magnification of 60. That is untrue. It's the product of both of them, so it'd actually be 10 times 50. So that one's out. Next, scanning electron microscopes and laser scanning confocal microscopes can both produce three-dimensional images. That is indeed true because they do it across the surface and scan like that and they get the contours. The maximum resolution of a scanning electron microscope is lower than that of a microscope. That is incorrect. It's a higher resolution actually because of the wavelength of the electrons being a lot less than light. And then also, transmission electron microscopes and laser scanning confocal microscopes can both produce three-dimensional images. So transmission don't, because it fires it at the surface. All they can do is just show you, like, where the electrons have been smacked at, basically, whereas these scan them across like this, and you can get those nice contours. So D is incorrect. So it must be B. So the figure shows a drawing of a light microscope image. The image is a cross-section taken from the stem of a dicotyledonous plant. This figure shows the diagram of a light microscope image. The image is a cross-section taken from the stem of a dicotyledonous plant. So here we've got F, E, and G. We need to figure out what all of those bits and bobs are. So it says which rows correctly identify the names of the tissue, E, and the functions of tissue F and G. So if you look at where E is sandwiched between these two things. So E, this is when you have this type of tissue between what looks like the phloem and the xylem, because these are tubes, remember, you can see these are the tubes, the xylem and the phloem. So then that's your phloem, that's your xylem, because these are bigger in on the outer part, and this is the smaller on the inner part. So between them, is a type of tissue, meristem stem cells, called the vascular cambium. So that means E is the vascular cambium, so it's neither of these two, that one's out. So then we need to think about what F and G are doing. Well, F is the phloem, so that's transport of assimilates, and then G is the xylem, so it's got to be that one, it's got to be A already. The other way around, so here, this is the opposite way around to what it should be. Which of the following statements A to D does not correctly describe the structure or formation of plant vascular tissue? All this question's asking is describing the structure or formation of plant vascular tissue, which means all it's just saying is, which part of these are wrong when it comes to describing the xylem and the phloem, because meristems differentiate into the xylem vessels. They become, so the stem cells and the meristems which become the vascular cambium, which eventually become the xylem and phloem. So it's just asking what's wrong about the description of these with the xylem and phloem. So companion cells are linked to the xylem by plasmodesmata. That one is incorrect. The companion cells are linked to the phloem, so that one's already incorrect. And so it's A. Mature sieve tube elements do not contain nuclear. That's correct. Phloem and xylem form differentiated by vascular meristems. That's what I just said before. Xylem vessel of non-lignified pits to light blue minutes. So it is A. All the other ones are correct. Large multicellular animals need a transport system for oxygen and carbon dioxide. Large multicellular plants do not need a transport system for carbon dioxide and oxygen. Which of the following statements A to D explains these observations? So if we have a look, then it says large plants have a low surface area to volume ratio. Well, no, that would mean that they would require one, because if you get a thin sheet of ice, for example, right, and you've got like an ice cube, and they're both the same length and height, just their breadth is different, this one's going to melt quicker because it has a higher surface area to volume ratio. So that means the movement of substances in and out can be higher. So if they have a low surface area to volume ratio, then that means that they're going to need a pretty fat transport system to be able to get things around the body. Because imagine you're trying to get something through across this thin surface. That's easy, but if it's quite thick, it's got to get all around it. So the only way around it is to have a circulatory system. So A is out. Then it says plants have a low metabolic rate. Well, yes, because we have a large transport system of the lungs and the heart and the circulatory system across the two of them. We need that to get oxygen around because we're moving, we're respiring constantly. The plants are literally just chilling there doing not very much. So it's B. There you go. Plants generate ATP during photosynthesis, so they do not need to respire. No, because the ATP is used for the Calvin cycle, etc., and it's not enough to keep them going. And then plants generate oxygen during photosynthesis. That doesn't really explain anything because they will then use it for photosynthesis as well, but that doesn't explain why they don't need a transport system, so it has to be B. Which of the following statements A to D describes the movement of water across plant roots? So the Casparian strip blocks movement by the Symplast pathway. So just as a quick recap, water moves through the roots into the xylem via two different routes, the Symplast pathway versus the apoplast pathway. So Symplast is living parts. What it means is that you've got cells that are connected to each other through plasmodesmata, and it's the water moves through the cytoplasm via all of these through osmosis. The apoplast is where you have the non-living parts. So what happens is that the cell walls, just water just can move through them and through gaps between them as well. So if it's in the apoplast pathway, then what happens is it will get to the endodermis cells in the roots, and there's a waxy strip called the Casparian strip, which basically forces it. So what happens is you've got like this water, it hits this strip, and then it's got to go into the Symplast route. So basically what it means is this Casparian strip stops the water moving, so it forces it to go that way into the Symplast route, and then it moves through cells and so forth. The reason that's quite important is because it means that water can go through the cell membrane, which are partially permeable, and that means you can actually start to control whether or not substances go through. Because if we just let water infinitely move through just via, standard osmosis through your apoplast pathway, there's nothing that can really control what's moving through and in and out of the cell. So 6 says which is the one that correctly describes the movement of it. So the Casparian strips blocks the movement via the Symplast pathway. No, it doesn't. It's the apoplast pathway, the dead pathway. The Symplast pathway requires water to cross partially permeable membranes. That is correct, because they need to go from cell to cell. So that's B. Water moves from the soil to the root held up a water potential gradient. No, they don't. They move down a water potential gradient. And then water moves through the leaves only by a Symplast pathway and across the roots only by that. Well, that's incorrect as well now. So now it says, which of the following statements about water transport in the plants are correct? So transpiration happens as a consequence of the need of gas exchange. That is indeed correct. It's where you lose water because the stomata are open. There are cohesive forces between the water molecules because they form hydrogen bonds with each other. That is indeed correct. Water is drawn up the stem due to adhesive forces between molecules. Well, no, it's cohesion that does it. So, and tension as well. So they stick to each other, but then the tension is what's pulling them up, not the fact that they're sticking to each other. So it's only 1 and 2, so it's got to be B. So the graphs A to D represent gas exchange in bony fish, and they show the oxygen saturation in blood and the lamellae in the fish, and the water flowing over the lamellae. So one line is water, one line is blood. So then it says, which graph A to D shows the relationship between the blood oxygen and the distance across the lamellae? So remember that this is the percentage saturation with O2, and this is the distance across the lamellae. The fish have a countercurrent system to make sure that the blood is always lower than the water, no matter what. So across the entire length of the lamellae and the gills and so forth, oxygen is always lower than blood. So it can't be this one because blood is higher than water in the terms of the saturation. So actually oxygen move out of the blood into the water, so that one's out. Here, water is always higher than blood, so it could be this, and it looks quite happy. Here, the water's dropping, and then the blood's dropping until they eventually reach equilibrium, so there is no more diffusion of oxygen between the two of them, so it can't be that one either. And here as well, water is always higher than the blood, so it could possibly be this one as well. So that one's out, this one's out. So now we need to look at the other part, which is water and blood moving in which direction. So I just said countercurrent flow means that this water is always against blood with a higher O2, so it's countercurrent, meaning it's got to be the opposite way. So that one's out already, so it's got to be B. So questions 9 and 10 refer to the graph below. An extract of the bark of this Indian fig tree, whatever, has antioxidant properties. It's been suggested that this extract can protect against non-cancerous cells during radiotherapy, so damage to them, sorry. Cultures of non-cancerous cells are exposed to increasing doses of radiation with a percentage of cells measured with experiments and performed multiple times with or without the bark extract. They show the results of the two sets of data. Each data point has one plus or minus the standard deviation. Fine. So the standard deviation is the measure of your spread of results around the mean. So can you see how all of these are in the middle? That gives you a hint that it's the standard deviation and it's the spread of the results here. Fine. And then we can see that without extract, as the dose goes up, the percentage that showed damage goes up as well. With extract, it can go up as well. So do it line by line and then between each other. So with it, as expected, the dose is higher, this goes higher. But then if we look without the extract, it's pretty high and steep. It goes sharply higher, increases. With extract, it sort of kind of goes up slower than this. So with extract, it's increasing more slowly compared to this, but it also doesn't hit as high of a point. The next part as well is to look at the overlaps and the deviation. So if we look at the without extract part, each one of these do not overlap. So they're all significantly different, meaning that like the results of this one don't overlap with here. With extract, you can see there's quite a lot of overlap between the results. In fact, all of them overlap, so we can't really say that this is significantly different to this, or this, or this, or this. So then it says, a student used Spearman's rank correlation coefficient to test for the correlation between the dose and cell damage. Which statement about this correlation of the graph is shown as correct? So correlation means as one goes up, the other one goes up, the other one goes down, the other one goes down. So if the correlation coefficient is 0, then there's no correlation. There's just dots all over the place. There's no pattern between the two of them. If it's, positive, it's greater than 0, then it suggests it's going up. So as one goes up, the other one seems to go up. And if it's negative, as one goes, the other one seems to go down. So here, Spearman rank correlation coefficient for cells without extract will be between 0 and +1. So without extract, it's going up as 1 goes up, so it's got to be that one. Wait, whoops. It's going to be that one. Whoops. 0 and -1 means it's going to be going. It's a negative result, so it's going to be going down, so that's incorrect. There's a negative correlation, nope. There is no correlation, nope. Because you can see as one goes up, the other one tends to go up as well, so there is definitely a positive correlation, so it's got to be B. Next it says, which of the following statements is correct? So the data with the extract group is more variable than without the group. So what that means is the variable is just how much spread you have around the mean. Ideally, a perfect result would be that this mean is just the same as its spread and there's no spread around the results of the mean. Whereas here, you could see there's the mean result, but you can see there's some that are higher, some that are lower, right? And the range is from whatever that is, like 35 to 40. But if we look here with the extract, there's a pretty, here's the mean, there's a pretty fat spread there from like 25-ish to 15. So there's a lot more variance in the results, as in there's a lot more variability. So for 10, the data with the extract group is more variable than without. It's got to be A. The error bar show the range of data at each thing. No, it does not. It shows the spread around the mean. The range could be something like this, where it's the highest and the lowest, and there's your mean. It doesn't always, if it's in the middle and it's told you it's, well, standard deviation, it's the standard deviation. So it doesn't tell you the range, it just shows you how much spread there is around the mean. As well as that, the errors bar show when the difference between the two is significant. So it's not the difference between the two sets of data, it's the difference between each measurement here is significant, not between the two. And then the chi-squared test can be used to determine whether there's a significant difference between the two sets of data. No, because we're going to be comparing the means, it's going to be the t-test, sorry. The chi-squared test is to measure its significance in the difference between the observed and expected variable. So if I flip a coin 10 times, I'm expecting to get it 5 heads, 5 tails. Maybe I might get 10 heads all and 0 tails. I'd use the chi-squared test to see what I expected versus what actually happened. So questions 11 and 12 refer to this investigation below. They should use a respirometer to compare respiration in 2 types of germinating seeds. Respiring seeds, soda lime absorb CO2. The starting position of the meniscus is here. Fine. What they did is they put a narrative containing coloured water. The position of the meniscus was noted at the start. They left it for 20 minutes, and then they measured the new position, and then they repeated it with different types of seed. Which of the following would be necessary to ensure valid results? So in these kinds of things, think of your independent and your dependent variable. So independent is what you're changing. So in this case, they are changing the seeds. The dependent variable is obviously the respiration, blah, 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 but as in what are they actually measuring here? It is the meniscus start and end. So what are they actually changing versus what are they actually seeing and measuring as a result of the change? Not necessarily just, respiration and germinating seeds. Like, just think specifically about these two, because these are the only two things that are allowed to change. Everything else must stay constant. So therefore, which of the following is necessary to ensure valid results? Keeping the respirometer in the dark during the experiment. Well, some seeds might need light to start actually doing germination, so that one's out. Keeping the respirometer at the same temperature, well, that is indeed correct. We need to control absolutely everything, and if we change the temperature, that could affect the starting point of the meniscus, or it could also change the respiration rate as well, so that screws it up. So with all of these, you need to think what could screw up these independent and dependent variable things that we're looking for. What could make this go different or this? And then it says using the same dry mass of seeds each time. Well, you should do that, because if we have more or less, then that can screw up this movement, and it can also screw up the amount of respiration happening as well. So 11 has to be 2 and 3, which is C. The students investigate 2 types of seed, pea and sunflower, and then pea stores mainly starch, and sunflower stores mainly lipids. Which of the following A to D describes the results you would expect with each type of seed? So, the meniscus would move to the left with pea seeds and further to the left with the sunflower seeds. No, because these are going to respire. They're going to take in O2 and they're going to release CO2, but then that immediately goes into the soda lime here. So if that's the case, then this meniscus should go that way, because there's a net loss of air volume because the CO2 is being absorbed the moment it's been taken out. So it can't go to the left because that's implying there's more gas being made that way, so no. The meniscus would move to the left of the sunflower seeds. Well, no, it would not move to the left. The meniscus would move to the right with pea and then further to the right with sunflower seeds. That must be the case because these were comparing starch versus lipid. Well, lipid can store more glucose per unit volume than starch. So if they're breaking down the lipid into glucose, they're going to be respiring more and so forth, blah, blah, blah. So that means they're doing more respiration, so they're going to move more to the right, so it's C. The meniscus would not move is incorrect because they're both going to eventually make glucose and then be used to actually, respire. So it's got to be C. So which of the following statements, A to D, describes the relative yield of ATP in anaerobic and aerobic respiration? So anaerobic respiration produces less ATP per molecule because glucose because lactate is converted to pyruvate, the other way around. Pyruvate's converted to lactate, so that one's out. Pyruvate's made from glycolysis to go into the link reaction, which then goes, makes acetyl-CoA, which goes to the Krebs cycle, and then you get oxidative phosphorylation. If it can't do that in anaerobic respiration, the pyruvate can't go into the link reaction, then it gets converted into lactate, cytoethanol in plants, that one's out. Anaerobic respiration produces less ATP per molecule because NAD is not regenerated in oxidative phosphorylation. It is B, because there is no oxidative phosphorylation happening in anaerobic respiration. If we look at the other ones, anaerobic respiration produces more ATP. Nope, it does not. Anaerobic respiration produces. Well, no, it does not. So it's B. So 14 and 15 refer to the diagram below of a synapse. We've got like, your vesicles containing a neurotransmitter. They release a neurotransmitter with a bunch of receptors and so forth. Fine. So which of the following statements, A to D, describes the events occurring at a synapse? So all synapses, remember, are just connections between nerves and another nerve or a muscle or a gland, some sort of effector. So in this case, what happens is an action potential arrives here. You get an influx of calcium ions, which cause vesicles containing a neurotransmitter to fuse with this membrane here. The presynaptic membrane, it releases whatever neurotransmitter is that it wants to use, and then it diffuses across, and then it binds to the postsynaptic membrane's receptors. This causes ligand-gated sodium ion channels to open, and then Na+, goes in, and then you get an action potential if the threshold's met. So here, let's have a look what's going on. So acetylcholine is broken down by enzymes so it can bind to structure K. That is incorrect. The enzymes break it down to stop it from staying here and get retaken back up in here. So that's out. An action potential causes J to close. That is incorrect. An action potential coming here means calcium ion channels open so vesicles can do its job. And then, then it says structure J is a voltage-gated calcium ion channel. Yes, it is. Structure L is released by exocytosis. No, it's not. It's not the entire thing. It's this stuff inside it. Those just vesicles, so that's out. So it's C. Next, it says GABA is a neurotransmitter. It reduces the number of action potential of the postsynaptic neuron by opening chloride ion channels in the postsynaptic membrane. Use what the question's telling you. So it says, which of the following statements, A to D, describes the action of GABA? So it has told you that you're putting Cl minuses in here. So if you got Cl minuses in here, what you're doing is you're making the membrane potential even lower, more negative, so you're hyperpolarizing it, meaning that you're going to need more Na plus to depolarize it. So it says GABA binds to structure K in competition with acetylcholine. No, it does not. It's literally told you it just does not do that. GABA causes hyperpolarization of the postsynaptic membrane. Well, I just said yes, because the chloride ions are moving in, making it more negative, so you need more depolarization to do, this, to get generated potential, action potential, and so forth, so it's B. GABA causes depolarization of the postsynaptic membrane. No, it does not. GABA inhibits the release of the neurotransmitter from that. No, it does not, so it is B. So figure 16.1 shows the structure of ATP. Name the circle component in 16.1. So adenosine triphosphate. So triphosphate, ribose, adenine. And also, as well as that, you know that you can identify it based on the fact that it's a purine because it has two carbon-nitrogen rings joined together, which you can see is the case here. So name the type of reaction that occurs when ATP is converted to ADP. So it's breaking down a molecule using water, so it's hydrolysis. A teacher told the students that the human body makes the equivalent of his own mass in ATP every day. Explain why, at the end of the day, only a small proportion of the student's mass was ATP. So be careful to just read the question carefully. It's saying that they're making like its own mass of ATP, but the question's told you at the end of the day that the small amount of their mass is the ATP that it's made. What they're trying to get at is that it's made and is broken, it's made and is broken, it's made and is broken. So that means that ATP is constantly hydrolyzed and then regenerated. That doesn't explain why we're doing that. It's so that ATP provides energy for, metabolic reactions. I don't know, like whatever you fancy. And it's a short-term energy source. Like, it's not used as a storage molecule. It's constantly being remade and broken. So they're saying that their body makes the equivalent of their own mass in ATP every day because it's constantly being broken down and made. And it's told you in the question at the end that if you picked like a certain time point, then the amount of ATP in their body is quite low compared to the rest of their mass. It's not saying that the teacher's wrong or there's something going on. It's literally just trying to get at the point that you're making it constantly and it's breaking. So student investigated the uptake of two different substances in cultured cells. The rate of uptake was measured at different concentrations of each substance in a medium surrounding the cell. So just think of your independent and your dependent real quick. So your independent variable is your concentration of substance. And then your dependent is rate of uptake. And the results are shown here. Look, just take a second to quickly describe to yourself what's happening one by one and then between each other. So here, as the concentration goes up in substance A, the rate of uptake increases and it's going through 0 and it's a straight line, so it's proportional. Whereas in the second one, the rate of uptake and the concentration as you see it increases and then it starts to level off and become constant. So you could say it plateaus, but you need to hint to the fact that it's becoming constant. People just say plateau and leave it there. That's not really enough. So if we compare A versus B, we can see that here the line is a constant straight line. How much the rate is increasing is constant, whereas here the rate is going up sharply and then it starts to level off. The student concluded that one substance entered the cells by simple diffusion, the other one by active transport. Evaluate this conclusion. So diffusion versus active transport. So if we look here, the rate of uptake concentration, as the concentration increases, the rate of uptake increases. So for this A, what we're trying to say is that the rate of uptake is proportional to the concentration. So as one goes up, the other one does. That is clearly got to be the simple diffusion. Because simple diffusion depends on temperature, the concentration gradient, surface area to volume ratio, and the diffusion distance. Now here, if you've got a really, really high concentration, the concentration gradient's really, really steep. So therefore, if that's going high and the rate's going higher, it must be that it has to be simple diffusion. So if you look at B, what's happening is that the rate increases, then plateaus, and becomes constant. So if we're doing active transport requires proteins. It's not that there's not uptaking things anymore. It still is. It's just it can't go any faster. It's just stopped there. So therefore, what must be the case is that the active transport carrier proteins or whatever, so the proteins must be saturated. And there you go. Because it's because of that. Because if we think about it, let me think. So for example, if we think about it, if I have like a cell here and I've got these channel proteins or whatever you want it to be, I've got two things can come in. It doesn't matter if I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. At any given point, only 2 can come in, so these are saturated, they're filled. Whereas if I just have something moving in by simple diffusion, it does not matter how many here and here. What's determining is the concentration gradient and all these other things I said before, so it can just freely move in as much as it wants. So clearly B must be that, because it's the concentration's so high, there's no more binding spots for these active transport carrier proteins, to work. So the student then added 2,4-dinitrophenol to the cells, and this inhibits respiration. Predict the effect of the DNP on the rate of the uptake of each substance. Well, we just said that substance A is a simple diffusion, so there is no effect. Why? Because it is simple diffusion, and that it is passive and does not need ATP, which is what the respiration's point is. It's to make ATP. For B, what must be the case is that it is reduced because ATP is required for active transport, because it is active transport, because it is an active process. So the student used a colorimeter to measure the concentration of each substance in a liquid surrounding the cells. The colorimeter had an analog display. The reading was indicated by a needle moving across the scale, so the needle literally would just wiggle like that, side to side, and the smallest divisions was 0.1 absorbance units. After the investigation, the student suggested some improvements. Draw a line between the improvement and its corresponding justification. So it says, use a colorimeter with a digital display to show absorbance to 3 decimal places. If that is the case, then it means you can distinguish between 2 spots better, because this thing can only tell the difference between 0.1 to 0.2. If I did it to 0.123 to 0.123, you can see that there is a lot. I'm able to tell much more precise differences apart. So that is resolution. It's the ability to tell apart 2 very close points and anything. So for example, if I draw these 2 circles here like that, if I zoom out really, really, really far, it looks like they're touching, but if I zoom in really, really, really far, you can tell they are separate. So you can tell these 2 apart, means you have a higher resolution. So in this case, it's got to be to increase resolution here. Next it says, check the 0 value with purified water before use. So if we didn't do that, so with these kinds of questions, just think what would happen if I didn't. If I didn't, then it would always add a separate value. So I don't know, if it started at 0.2, and every single measurement I had would have a 0.2 added on top. When you have an error that's affecting everything the same way in a systematic sense, it's a systematic error. So there you go. Now, for each concentration, repeat the measurement 3 times and calculate a mean. The reason is because if we just did, let's say I measured it and it was 0.6, and then I did it again and it was like 0.9. I'm not really confident it's lying around 0.6, but if I do it again, it's 0.5. Now I'm like, okay, now I'm a bit happier that it's around the 0.5 to 6 mark. So the reason we do that is to reduce the impact of random errors and uncertainties, which would be here. And then the last one says, ask students in several schools to carry out the same investigation. It's so that we can look at whether they're able to get the same experiment done and get the same results, but it's different people. So repeatability is the same person doing the same experiment and getting the same results. Reproducibility is you can give them the instructions, they can do it, and they get the same thing. So it's here. That's what it is. So student investigated the photosynthetic pigments and spinach leaves using thin layer chromatography, and they showed the plate at the end of this investigation. So origin, solvent, front, whatever, right? So what you do is you dip these in the solvent below the origin, and what happens is that the solvent will move across all the way to the solvent front, and these will all move in different things depending on how long they spend in the mobile phase, the moving phase, versus stationary. So stationary phase is the plate itself where it's stuck. Mobile phase is the solvent moving across, it's moving. So if you're more soluble in the mobile phase, it means you will spend more time in the mobile phase, meaning you're going to move further if you're more soluble in it. If you're less soluble in it and better adsorbed, you're sticking to the stationary phase, it means you're going to spend more time stuck to there, so you end up moving here instead. That's always helpful to know. The table shows colors and RF values for these pigments. The RF value, remember, is just the distance of the spot over the solvent front. So then it says calculate the RF value for spot 3 and then identify what was used. So all you would do is you would just work out the distance of the spot from the origin and then the origin to the solvent front, divide the two, right? I'm just going to, for the sake of time, just go ahead and just jump ahead. So it's 0.53, so the pigment must be chlorophyll A. That's all you got to do. Just to save time, there's no point in me sitting here awkwardly measuring it. So then it says predict the color of spot 4. So if we look at spot 4, well, we know that this one now is chlorophyll A, which is here. Spot 4 has moved further, so it means it has a higher RF value, but it's not highest because 5 is the highest. So it's the second to highest RF value. So if we look at what our options are, the highest RF value we can get is here, and then the next one is this. So that means that spot has to be gray. So now it says the solvent used for the separation was non-polar. Identify the spot corresponding to the least polar pigment and give a reason for your choice. So the solvent, remember, is the mobile phase. So if it's a mobile phase, remember, if you're more soluble in it, it means you move further. If I dissolve, or if I mix oil plus water, they do not dissolve. Because the reason is because oil is non-polar and water is polar and they don't like each other. If I mix water and, I don't know, some sort of acid like HCl, it will dissolve because they are polar and polar. If I mix an oil and another oil, like a fat of some sort, they will dissolve because non-polar and non-polar can dissolve with each other, they can mix. So in this case, they've told you that the solvent that they have used is non-polar. So if it's non-polar, then the thing that moves the furthest is also going to be non-polar, because remember, if polar and non-polar don't like mixing. So if it's asking you what the least polar thing is, it means it's moved the furthest, because the less polar you are, the better you're going to mix with another non-polar thing. So in this case, it's got to be spot 5, which has moved the furthest. Why? Because it is the most soluble in the mobile phase, because the mobile phase is non-polar. Non-polar likes non-polar. So the student did this following method for this investigation. They extracted pigments by doing all of this. They did, so actually no, let's read it properly, shall we? So they took 0.5 grams of fresh spinach and 1 gram of salad, ground it until they got a light green powder, transferred it to a test tube, added propanone, stirred it for a minute, and then allowed it to sit for a minute, transferred the green upper layer to pipette with a clean test tube, and then sealed the film when not in use. And then what they did is they hold the TLC plate carefully by the edges to avoid damaging the surface. They draw a pencil along it with 1 centimeter from the bottom edge. They spot the extract using a pipette one at a time, allowing it to dry before adding the next one. They place chromatography solvent in a jar so it's no longer than 0.5 centimeters deep. They lowered it into the jar and then leaned it the top against the jar, made sure the plate does not touch the sides of the jar anywhere else, and then they placed a cap on the jar, allowed it to soak up the plate. When the solvent was reached a few millimeters from the top, they removed it from the jar and then marked the position of the solvent front with a pencil. Fine. So said explain why they did the following precautions. They held it carefully by the edges and avoid damaging the surface. Well, the reason is because the movement depends entirely on, the substances moving across the plate. So if you've damaged the plate, then you're going to screw up the movement. So it's to avoid affecting movement of spots. Then it says to make sure the plate does not touch the sides of the jar anywhere else. Well, if it did that, then what if there's like condensation on the jar or something like that? Well, there might be liquid on there because the stuff that you're putting in there is going to be evaporating and condensing there. Also, you could say something like, what if it it touches the jar on the side here, and then this spot just can't move anymore because of this stuff interfering, like on the side, like the jar touching it. So then, in that case, I'm just going to go ahead and say something like, condensation of the jar may affect movement. In this question, what I'm thinking is the only thing we're measuring here that we want is the movement of the stuff in the solvent front. So any precaution is probably going to be doing something to avoid that element being screwed up. Now it says to suggest an advantage of working as quickly as possible in step 1. Remember that this experiment is all about looking at photosynthetic pigments in a plant. So if they're saying to do this as step 1 as fast as possible, or extracting them as quick as possible, so the only thing that comes to mind is, well, what happens if they take ages? So what they've done is they've been basically breaking it all out and releasing it into stuff, and also putting propanone in. So to my brain is saying, well, okay, if they're doing it as quick as possible, then it's probably to stop, something happening to the pigments, like getting them damaged too much, for example. So to avoid, you could say damage, degradation, or something like contamination, things like that. So it could be to avoid damaging the photosynthetic pigments. But also, as well as that, the questions told you that they have put this propanone in here as well to allow it to, do its job. If they left it for ages, maybe the propanone is, because that's the solvent, remember, might evaporate, and that could screw up the solvent front as well. So algae are single-celled photosynthetic organisms. An investigation was carried out to see the effect of CO2 on the concentrations of ribulose bisphosphate and glycerate phosphate. So your independent is what? We're changing CO2 concentration, and your dependent variables, there's two of them, is going to be RUBP and GP. Just always good to think that in your head. So algae were grown in a solution containing a high concentration of CO2, and then they reduced it slowly, and then they looked at the RUBP and GB concentration for 2 minutes, and then 3 minutes. So 2 minutes before, 3 minutes after, and the reduction of the CO2 concentration. The results are shown here. Fine. So again, one by one, look at each line and just make sense of what's happening. So the first 2 minutes before they've done what? So if you look line by line, this is GP. The concentration of GP is constant, and then once they reduce it, it seems to drop. And then RUBP is constant, and then once they reduce the CO2, it goes up. Now I'm going to do lines together, because if you do it one by one, it means you don't miss anything. So now I'm going to compare the two. So to start with, GP is higher, RUBP is lower, but they're still constant. And then dropping the CO2 means GP drops, but RUBP goes up. But then RUBP goes up, peaks, and then starts falling back down again, right? So if you do it like that, it means you don't miss anything. So then it says, describe and explain. So describe is say what you see on the graph and then explain why it's happening. So if you look at describe and explaining the effect, then let's do it one by one. You should always do it one by one. So describe and explain. So if we look at GP, decreasing the CO2 decreases GP, and then we need to think why. So RUBP, remember, combines with CO2 using Rubisco to make 2 lots of GP, which eventually becomes 2 lots of TP, and then is eventually regenerated back to RUBP. So clearly, if we're decreasing CO2, that makes sense. One sec, redo. So less CO2 shows that there's less GP because there's less RUBP combining with CO2 to produce GP. So that explains the GP curve. Now if we look at the RUBP curve, what happens is then it increases and then decreases. It's because, well, we need to think, why is it increasing? Well, because less conversion to GP, and then we need to explain, well, that's why would it go up though if it stopped? Because it's a cycle, right? So it clearly must be there's some TP remaining which produces RUBP. However, as well as that, we've not really gone into why it's decreased. So we need to think why it's decreased. The only thing that lets it increase must be the fact that, GP eventually regenerates TP and so forth. So less GP means less TP, which means less RUBP regeneration, and therefore it drops as well like that. The next question says that FeRA2 is a fluorescent dye that can be used to measure the concentration of calcium 2+ ions inside cells, and the structure is shown below. Here we have it. It says use information in the diagram to explain why it cannot cross cell membranes. So cell membranes, remember, are your phospholipid bilayer with lots of little bits and bobs and proteins and stuff embedded within them and whatnot, right? This middle part, you have a hydrophobic bilayer, which means that because it's hydrophobic, polar things can't move through. Now here in this, it says use the information in the figure to explain why. So we need to look in this figure to see what's stopping it from coming through. And as you can see, here, by default, it's polar straight off the bat. So that's exactly why it can't come through straight away. So it's because it is charged, therefore it's hydrophilic, so it cannot pass through the hydrophobic bilayer. Additionally, if you wanted, you could also say that it's a pretty big thing compared to glucose, which is like one singular 6-carbon thing. Or even then that can't get through, actually. Or like water, which is teeny, and oxygen. This is huge. So you could talk about size as well. So scientists have used FeRA2 to study the role of calcium 2+ ions in the synapses of living nerve tissue. It was injected into a single sensory neuron that was connected by a synapse to a relay neuron. FeRA2 fluoresces inside the neuron, and it was observed using a confocal microscope. So just one advantage of using a confocal microscope in this study. So a confocal microscope is a special type of light microscope. And what it does is it essentially points a laser beam and moves it across a surface. And there's usually like some fluorescent dye tagged, which in this case they're using that on the specimen that you're testing. And what happens is the dye will fluoresce, it will emit light, and then the light is picked up back from a detector that passes through a pinhole, sorry. So pinhole will only let some of the specific light through, and then all this other light will bounce off to the sides and stuff, and it won't get picked up, and that gives you a really, really clear image. One of the advantages of a confocal microscope, however, is that it's quite good at using thick, like, you know, looking at things in thick specimens. So it's good for, so can be used in thick specimens. The sensory neuron was stimulated electrically, and the FURA2 fluorescence in the synaptic bulb was measured. At the same time, the electrode recorded the membrane potential in the postsynaptic neuron. The results of this table are shown below, so have a quick skim through it, as always. So strength of electrical stimulation, we've got low, medium, and high. So what happens to the fluorescence? It's also low, medium, and high. And then the highest membrane potential is negative, minus 60, and then it's plus 40, and then plus 40. Fine, and this is to do with how strongly it's being stimulated. The intensity of FURA2 fluorescence is proportional to the concentration of the calcium 2+ ions. The scientists concluded that changes in the concentration of these ions, these Ca2+ ions, in the presynaptic neuron cause an action potential in the postsynaptic neuron. Explain and evaluate this conclusion. So remember that an action potential is where you've got depolarization that's happening and then it peaks, sodium ion channels close. There's only one way to get the charge to go back down, to repolarize and become negative again inside, and that's by opening potassium ion channels like this, bang. And then you get the overshoot, and then you've got your action potential, and it rinses and repeats. The reason sodium moves in is because if you remember in your resting membrane potential, you've got 3Na plus out and 2K plus in. So because there's now lots out, that's a high concentration of sodium outside and very little in. So that's why you get that massive influx of it to start with. And then potassium, because you've moved it in, you've got a lot of potassium inside a cell. This is during your resting membrane potential, remember? So then when you're depolarizing, if we open sodium and let it carry on, it would just shoot off like that because, you know, sodium wants to come in because we've shoved it all out. So what happens in repolarization is potassium is moving from the inside out. Yeah, so there's that, so that's work. So here we can see that when we're stimulating it, we get from negative to positive, so there's depolarization. But when we go from medium to high stimulation, the maximum potential is not changing. So that must suggest that we've hit like the highest it will get, which means that must be, you know, an action potential, because all action potentials are the same size. Whenever you got these evaluate questions, it's always helpful to write obviously what they're saying leads to what, and that helps a lot. So they're saying that Ca2 plus changes led to an action potential. So remember that this represents calcium 2+ ion concentration. Low stimulus leads to low amounts of that, medium, and so forth. So if that's increasing, then remember that this is the presynaptic neuron that they're messing around with here, and synaptic bulb presynaptic neuron, and then the postsynaptic neuron is what they're measuring. So, well, first of all, we could start by saying that increasing the stimulation leads to an increase in FURA2 fluorescence. I'm just going to write fluoresce because I'm lazy, which signifies an increase in Ca2+. But then you can see that what's happening here is this increase in Ca2+ eventually leads to an increase in the postsynaptic neuron. Remember, presynaptic's got more calcium, and then eventually this postsynaptic neuron has an AP in it. So the only reason that must have happened must be that Ca2+ has entered, your vesicles are fused here and release neurotransmitter like that. So we're going to describe that. It's increasing Ca2+, meaning that Ca2+ causes fusion of vesicles containing a neurotransmitter with the cell surface membrane. It's released into the synaptic cleft. You know the whole steps, diffuses across, binds to receptors on postsynaptic neuron. Na+ enters the postsynaptic neuron. You get depolarization, which is why it's going, you know, you've got an increasing membrane potential, and then eventually you get an action potential because it's staying constant at plus 40 millivolts. Remember that action potentials are all the same size. Sorry, and I should really say if the threshold's met. Now, these scientists are saying that the concentration of this in the presynaptic neuron leads to an action potential in the postsynaptic neuron. We've just described all these in-betweeny steps. We can't definitely say that, you know, the changes in concentration of Ca2+ leads to this directly, because we, you know, like we're only just noticing these are linked, not that they're definite causes. So therefore, some, you know, limitations are that the increasing Ca2+ may not solely determine the postsynaptic membrane potential, and we can see as well that it might not be an action potential. It could, well, I mean, it is an action potential, sorry, but like, we can't say for sure because, you know, here they're saying that changes in this lead to a change in that. We've only gone from low, medium to high. We have no numbers on, like, you know, for example, what they are and stuff like that as well. And there's no membrane change between medium and high, so we might not have done enough there. Like, you know, results, they could have done, like, I don't know, medium high or something like that, or low medium, and there could have been numbers in between. So then, from medium to high, there was no change. So we could consider using some more numbers in between. The image below is a photomicrograph of a mitochondrion. Name the type of microscope used for this. So we can see the internal structures already. So that's got to be one of the electron ones, and it's a two-dimensional image. So I'm going to just write transmission electron microscope, but I'm lazy. You should write the whole thing out. Identify the structures labeled M and N in this microscope. So the M is pointing at this juicy, you know, stuff between the folds, which is going to be your matrix. And then N is those folds of membrane, which are the cristae. Crista or cristae, that's fine. So then it says, describe 2 ways in which the structure of the mitochondrial membranes is related to its function. So it's asking about what is in those membranes. So one of the things we could say, first of all, is that they've got, you know, like ATP synthase channel proteins in them. And then it says related to its function. That's important for oxidative phosphorylation. Oxidative phosphorylation is ATP synthesis. I should have written it the other way around. ATP synthesis in oxidative phosphorylation. So that's one. What else have we got? Well, it's also got the electron transport chain. And that's important because it's the decreasing energy. So the decreasing electron energy is used to create a proton gradient. It says mark as continuous prose, so make sure you just really write these in full sentences. I'm lazy, so I'm doing it this way. The endosymbiosis theory suggests that mitochondria may have evolved from bacteria that were taken from other cells. These cells then evolved into eukaryotes, give 2 structural features of the mitochondria that supports this theory. So this theory is that, you know, back in the day, these were like chilling bacteria or prokaryotes just having a great time. And then what happened is another cell was like, yo, I'm going to eat you, but I'm not going to digest you if you like make stuff for me. And if you make ATP for me, we'll be happy. So this guy was like, all right, fine. I'll do that for you. Here we are. So you need to look at this mitochondrion and look at what's similar to it and prokaryotes. So if we look at this, then some of the things that we can say, first of all, is that mitochondria contain, these are just features that you should know, they contain circular DNA. And then as well as that, one other thing that you can say is that they have those, it has ribosomes. And if you want to be specific, it's got 70S ribosomes. So that suggests that this could have been its own cell because, you know, the mitochondria could have been its own cell and then eventually was taken in. And also there's no nucleus of its own and no other, you know, like mitochondria within it itself and all that kind of stuff too. So that supports that. Other things you could have said as well is it could have plasmids, it could have, you know, its size is similar and so forth. But these are 2 that I'm thinking straight away. So now it says, explain why early eukaryotes were able to grow more quickly than cells that did not possess mitochondria. So all it's saying is that eukaryotes, so what it's saying here in this question, the question has objectively told you that having mitochondria means that you can grow faster. So then you need to think about why. What is the link? Well, the only link that it would be is that they can respire aerobically, which means you've got more ATP production. Therefore, there's more energy, because we're going for growing faster. There's more energy for, and then you need to name something. You can't just say growth. So like protein synthesis, for example, or you could say DNA replication. If you're going to say growth, you have to give it an example. So for energy for growth, for example, or e.g. protein synthesis or whatever. And there's also more energy for metabolic processes. That's it. So a student investigated the effect of different sugars on the respiration of yeast using this following procedure. So it's always really good to think of your dependent and independent variable, but also remember like to link it to what they're actually measuring and looking at. So what you change, what you measure, obviously you could argue, yes, they are changing sugars, the respiration rate in yeast, but let's actually narrow it down to what they're actually measuring. So if we look here, it says prepare a stock solution in yeast with dried yeast. They prepare solutions of sugar, so they're independent of their changing is the sugar. What are they doing? They're just containing, okay, so it is just different sugars, fine. Dependent is what they're measuring. So they keep the yeast to sugar in a solution of this until required. They then set up the apparatus with a conical flask, rubber tube, gas syringe, and then they added this and this to the flask, immediately connected it inside the clock, and then they recorded the volume of gas produced. So the dependent measure is the volume of gas, which they are using as a representative of respiration rate. So then it says repeat the experiment 5 times for each different sugar, and then prepare a fresh yeast solution for each set of sugars. Fine. It says, describe 2 precautions the students should take between each experiment to ensure repeatable results. The reason I said label this is because you need to now look at what can screw up both of these in the process. So look one by one and see what can screw up the sugars, amount we're using, the concentration, its volume, whatever, what you need to control with this, and what can screw up what we need to control about this. So first of all, I could probably go ahead and say, well, a precaution they could take definitely is to clean equipment between use. Not mention that. Or, like rinsing them stuff. As well as that, they're just slapping it into a thing and, like what if all the yeast is settling at the bottom and there's like sugar here, for example. So they should stir the yeast stock solution so you get even distribution. As well as that, what if, so they're saying immediately connect the gas tube and start the clock. What if they haven't connected it properly and it's not, airtight? There might be leaks in the syringe. So again, it's just looking at every single critique possible for these 2 things that can get messed up. The student's procedure did not contain a negative control. Describe 1 negative control that they should have used. So when measuring the volume of gas, our control needs to be something that should ideally make no gas. The thing that's making the gas here is the yeast. So we're going to use we're going to deactivate the yeast. So we need to boil and cool yeast. That way they are dead and you're not going to get any gas produced ideally. So again, labeling this really helps. The student then calculated mean values for each sugar and then plotted the results on a graph, and the graph is shown below. Describe how the student could use the graph to calculate the rate of respiration for each type of sugar. So just a quick idea of what's going on in this graph. Glucose graph, it's increasing and then it sort of kind of starts to increase less quickly and starts to level off. So that means, we're probably stopping, using up the glucose and respiring. It could be respiration stopped. It could be that we've run out. Lots of different reasons why. Maltose, similar principle. It's increasing and then it sort of starts to level off and then increases a bit slower and then faster and then levels off. And then here it's increasing slowly and then it increases more sharply and then levels off. They all, one by one each line and then with each other. So glucose starts off with the most gas and it levels off between maltose and sucrose. Maltose increases slowly but then increases more quickly and actually supersedes glucose, and then sucrose is the lowest of them all and it sort of increases slowly and then it increases quickly and then levels off at the least amount of gas produced. Fine. So because they want to look at the rate of respiration and they're using the volume of gas as something to represent respiration, rate is the change of something with time. So whenever you have a graph, the gradient as a pointer is your change in y of your change in x, right? If we look at the units, that's going to be centimeters cubed over minutes, which is going to be centimeters cubed per minute, which is a rate. For example, for those that, I don't know if you guys remember your physics, if you do distance over time, the gradient is going to be the change in distance over the change in time. Distance over time, so it's going to be velocity. And if you look at that in meters and that's seconds, meters per second squared. So the gradient of a graph is generally the change in y divided by x, so you can work out the units from that and figure out what quantity it is. And the area under a graph generally is the 2 times. So for example, if I have velocity and time, displacement or speed, equals velocity times time, right? Speed equals distance over time, so then distance equals speed times time. The area under the graph tells you the distance, so you can always use that as a pointer. So generally speaking, the area is the change y times x, and the gradient is the change in y of the change in x. Anyways, so back to this. It says explain how they can use this graph to calculate. So well, I can see on their y-axis it's volume of gas produced, and on the x it is time. And also, sorry, I forgot lactose existed. Lactose was untouched and only increased slightly. So then what they could do is for each sugar, what they could do is they look at each graph and they would draw a tangent at each point like this and then work out the gradient of the tangent. Because the steepness of a graph tells you the gradient. We've just said that the gradient is the rate, right? So if the gradient's steep here but not so steep and then it's steeper, it's changing. The gradient's changing, the rate is also changing. So what we would say is that we need to draw a tangent at each point, and then what you could do is you could then work out the gradient of the tangent, which is your change in y over the change in time, so change in volume of gas over time. And then what you can do is you could compare the rates calculated between each sugar. So here, if I drew a tangent here, that's a quite steep tangent. The gradient's quite steep, so there's the rate is quite high. Here, here it's not very steep. It's less steep, so that means the rate is slower. Here it's not very steep at all, and here it's like barely existent. So there you go. So the student concluded the following things. The rates of respiration with glucose, maltose, and sucrose were very similar. The yeast could not hydrolyze disaccharides. Evaluate their conclusions. So let's look at the supporting statements first. So they're saying the rate of respiration is similar. Remember that rate is a gradient, so they're saying the gradients are similar. Well, if you look at each curve, The gradients are quite similar, like amongst the most of it. You know, like in the middle parts here, they're quite similar, but the problem is that it's also changing throughout. So here and here and here, and they level off. So that's one piece of evidence to say that it does support it. So the slope, you know, the gradients or slopes or steepness, whatever you want to say, the gradients are similar overall. And then ideally, you could probably calculate some as well, too. I'm going to skip that out for now, but you should really calculate an example. So here, for example, and then here, work out the gradients of those tangents and then compare them. So then, you know, I'm going to write calculations to support this. Just for the sake of time, I'm just going to move ahead. If you want to know how to do the gradient, all you do of a gradient of a tangent is draw the tangent. You pick 2 points. You do the change in the y. So here, minus the change in the x, which is going to be y2 minus y1 over x2 minus x1. So your second y coordinate minus your first over your second x minus your first. So there's that. Then as well as that, they've said that these could not hydrolyze disaccharides. If we have a look here, what's supporting that? So, well, I mean, if you look at lactose, that's a disaccharide, and there's barely any respiration. There's barely any gas produced. So you could say lactose is a disaccharide, and there's barely any gas produced. So that suggests very little respiration. Therefore, yeast may not have the enzyme to hydrolyze it. Then so let's talk about some limitations then. So limitations at both points. The rate of respiration limitations, well, if we have a look, they're not consistent. So this sharply increases followed by all of them. So we need to think of an explanation. So glucose increases, you know, the rate is higher, sorry, initially, then the others catch up. So that suggests that respiration must be starting sooner as well. So then, as well as that, glucose increases more rapidly. The rate, sorry, then levels off. And then as well as that, you know, you can also see that, you know, as glucose is slowing down, these still increase. You can see that this is starting to drop off the glucose, but this is still increasing. So as the glucose rate drops, sucrose and maltose still increase. The reason I'm saying all these points is we need to think of an explanation why. It's suggesting that the glucose has had a head start, and that if these didn't have a head start, then they would be over here instead. So then what must have happened is, remember, glucose versus sucrose and maltose, disaccharides, it must be that, you know, the only thing I can think of is that we can only use glucose in respiration, so they must be hydrolyzed first. So that means that maltose and sucrose needed hydrolyzing to be used in respiration, so they lagged behind. So that's a limitation of the first point. Do it one by one. And then in regards to the what's it, the other one, which was the disaccharide bit, well, hello, these are disaccharides, and they still were able to do it. So this student got it wrong there, because maltose and sucrose are disaccharides and were respired, right? So it's not disaccharides. It might have just been that they were unable to do with lactose only. So maybe they lacked enzymes for hydrolyzing lactose only, or it couldn't absorb it. Like maybe it could be that it was, you know, because we can still see there's a little bit going on here, so they might not have absorbed very much. So, or couldn't absorb its products. That's me rinsing the data, but remember, when we're doing an evaluation, we need to look at their method as well. And also the results, can you trust them? I've written so much, let me just erase that. What I mean by that is, can you as a scientist look at this and say, yes, 100%, this makes sense, these results are great. I ain't happy with that personally, because there's multiple things going on here. First of all, I have absolutely no clue, because they've worked out the mean values of each sugar. I have no idea if any of these means are different. I have no statistical tests with them, so I don't really trust it already. Generally speaking, when we're looking at results in science, we like statistical tests. If there are none here, then we should point that out. As well as that, you know, they're talking about respiration. We've only measured the volume of gas collected. Like I said before, you know, that they're using it as a representative for respiration rate. That doesn't necessarily mean that this equals this, which is why you should always go back to just, you know, linking it back to that starting point. So some things there is that there's no t-test, or standard deviation is what I would use, because t-test is for comparing means. Standard deviation is looking at the spread between them. So there's no t-test, or there's no SD, so we don't know if differences, and make sure you say what the results are. The differences in mean rates are significant, and as well as that, volume may not represent the rate of respiration. And then that makes sense because, you're like, you're taking in oxygen and putting out CO2. We have no idea if it's aerobic or anaerobic. I think they mentioned it's aerobic, anaerobic. No, it was that too. So we're presuming it's aerobic. We don't know if there's any anaerobic stuff happening here as well, which could obviously get in the way of how much CO2 and O2 you're messing around with and stuff. So it's just really important to be cautious about that. So the student wanted to investigate the effect of different sugars on the rate of growth of a yeast population. So again, you're independent and you're dependent. I wouldn't jump straight into that yet. Look at everything first. They use the colorimeter to measure the absorbance of yeast culture cells. The absorbance of the yeast is proportional to the concentration of yeast cells. As the yeast multiplied, it was necessary to dilute the sample to obtain a reading on the colorimeter. So then, if we just have a quick skim of everything else, what did they actually do? So then, they did what? So their independent variable is the sugars again. Well, are the sugars, sorry. The dependent might be the absorbance from which they're deriving the number of cells. This may change throughout when we find out more about this experiment, but it's always good to just jump into there and have an idea of what to look out for, because anything that isn't this needs controlling, and anything that isn't this needs controlling, and they can all be screwed up by these two things, and it helps you go down what, to say, for example, precautions-wise, limitations-wise, and so forth. Describe how the student can use 1 centimeter cube pipettes and 10 centimeter cube measuring cylinders to dilute the sample so that it was 10,000 times less concentrated. So if we're doing that, we're using a 1 and a 10 centimeter cube. So if I get 1 centimeter cube solution and 9 centimeters cubed water, that means I have 1 and over 10 in total. So it's a 1 in 10 dilution. If I then do the same thing again with it, then I'm going to do a 1 in 10 dilution of the original 1 in 10 dilution. So what I'm actually doing is I'm timesing it by 1 in 10. And if I do it again, I'm timesing it by a 1 in 10. And if I rinse and repeat this, it will be a times 1 in 10 every time. So what I mean by that is that this is 10 times less concentrated, or more diluted. So if they're saying that this is 10,000 times less concentrated, we're going to do a 1 in 10, 1 in 10. That's going to be 1 in 100. So 100 times less concentrated. 1 in 10 is now, again, it's 1,000 times 1 in 10 again, 1 in 10,000, which means it's 10,000 times less concentrated or 10,000 times more dilute. So what you would do is you would add 1 centimeter cubed of the, culture, and 9 centimeters cubed of distilled water. To work out any dilution factor, you would just do the amount of what you used, the original stock, over the total volume. And that will tell you by how many times the concentration is less on the bottom. So again, like here, 1 in 10, 10 times less concentrated or 10 times more dilute. So we've done that, and that gives you a 1 in 10 dilution, and then what you just do is repeat it 4 more times. So repeat up to 4 times, and then you've got a 1 in 10,000 dilution, and then each time you should describe each step. So 1 in 10 to 1 in 100 to 1 in 1,000 to 1 in 10,000, and then also, as well as that, it says describe how the student should do this. We should also talk about experimental techniques too as well. So ensure you don't want to just have it all settle at the bottom and then take some out and miss all of it. So you want to ensure that you're mixing the yeast at each stage as well. So that way you are always making sure that it's evenly spread out. So when you take out 1 centimeter cubed, you know that you're taking the right amount out and you're not just taking out like the watery bits above it. A light microscope can be used to observe yeast cells. State the equipment that would be needed in addition to a microscope to measure the average diameter. So to measure them, you're going to need an eyepiece graticule. Or else how are you actually going to look at the line and sort of measure it against its stuff? And also you're going to need a stage micrometer for a scale as well. Then it says the student prepared a starter culture using 2.5 times 10 to the minus 3 grams of yeast in 1 decimeter cube nutrient broth. The average mass of a yeast cell is that. Calculate the number of cells. So 2.5 times 10 to the minus 3 grams of yeast in a decimeter cubed. And it's saying that 1 yeast weighs this much. how many cells is it going to be? It's going to be your total in this sample, 10 to the minus 3, 2 by 5 times 10 to the minus 3, or 2 times 10 to the minus 11. If we do that, we should get, and they want it in standard form. So if I look at my calculator, I've got 1, 2, 5, 0, 0, 0, 0, 0, 0. If you press the ENG button on a Casio calculator, the ENG button, it puts it to 125 times 10 to the 6, but they want it in standard form, so I'm just going to do 1.25 times 10 to the 8. So that's a quick shortcut and like cutting down the number of, significant figure decimal placey bits. You can put any number in a calculator, and if you press that, it will do it in threes. So 10 to the 3, 6, 9, and so forth. So you can use that to then work out the other two and, cut down the workload a bit. There you go. The population of yeast cells doubles every 90 minutes when growing under ideal conditions. A different starter culture is prepared using 1 times 10 to the 7 cells. Use the axes below to sketch the growth curve that you would expect for the first 15 hours. So we need to go through every single point, and it says here it's log 10 number of cells, so we need to log 10 everything. Now if I log 10 to the 7 in my calculator, so we're starting at 7. That's our first point. Now, rather than going through each and every single point, you could probably just shortcut it. 15 hours, we can assume that it's going to grow linearly if it's log as well. And because obviously, if it was just the raw number of cells, it would go up really high and really fast. So the log makes it a nice linear thing here instead, because it's just, 1 times 10 to the 7. If you kept doubling that, that goes huge. So log things let us fit, massive ranges of numbers on a graph. So 15 hours, that's going to be 15 times 60. That's 900 minutes, right? So that every division, it doubles every 90 minutes. So now we're going to have 10 doubles. So after 15 hours, we'll have 1 times 10 to the 7 times 2 to the 10. Times 2, 10 times 2 times 2 times 2 times 2, and so forth. So that means you're starting, so then the end is going to be this, which gives you what? 1.024 times 10 to the 10. So if I log 10 this, then I can find out where my last number is going to be, which is around 10. So the 15 mark is going to be around the 10-ish mark here, so then I can draw a straight line connecting the two like this. That's what we would expect. The table shows the features of the airways and the lungs. Complete the table by putting a tick in each box if each feature is present and a cross if it is absent, and each structure of the first row has been done for you. So in the bronchi, you have got cartilage, you have got elastic fibers, and you have got goblet cells. In the bronchioles, don't really have as much. We have elastic fibers, you don't have goblet cells, and in the alveoli, you don't have cartilage, you do have elastic fibers, sorry, because they have to, expand and recoil. And then you don't have goblet cells within the alveoli themselves. So there you go. That's just sort of memory, that one, unfortunately. So ventilation of the lungs creates air movements that can be measured and recorded. Name the apparatus to measure this. It is spirometer. Next. The trace below shows a recording of ventilation movements from an individual subject. So this is the volume of the air in the chamber. You can see it's going up, down, up, down, up, down, up, down, up, down. And there's a big down, and then a big up, and then a up, down, and so forth. So when it's going up, down, up, down, up, down, air is going in, air is going out, air is going in, air is going out. So that's them breathing and whatever, right? So then it says use the trace to estimate a maximum volume of tidal volume in the first minute. So tidal volume, so when you think of tides, they're coming in and they're coming out, they're coming in and in a beach, right? So tidal volume is the amount of the volume of air in every single breath you're taking, right? So in this case, we're going to be looking at the maximum difference between the top and the bottom until we find what we need. So the one that has the biggest gap. So based on that alone, maybe around like here to here, no, there to there, which is going to be what, 3.5 minus 2.4. If we do that, that gives you 1.1 decimeters cubed. Because remember, this is only measuring the volume of the air in the chamber before and after. So if it's going up, the person's breathing out into the tube. If it's going down, they're breathing back in. After 60 seconds, the subject was told to breathe in as deeply as possible and breathe out fully. Use the trace to calculate the vital capacity of the subject. So just be cautious. So this is the volume in the air chamber. So this is them breathing out when it goes up, not in. So after 60 seconds, blah, 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 blah. Yep, so if we look at where 60 seconds is, we're looking at the vital capacity. The vital capacity is the maximum volume of air that could be breathed in or out. So here, we need to look at the lowest point possible and then the highest point possible, because that's them breathing in as much as they, breathing out as much as they possibly can, because it's the volume of the air in the chamber. So the minimum point possible is 0.12, and then the highest point possible is like 4.7. So it'll be 4.7 minus 0.2, which is 4.5 decimeters cubed. So that's that. So as a reminder, just on normal spirometry curves, tidal volume is the volume of air in each breath. Remember that, think about it like this. Because this is in the chamber, if it's going down, air is out of the chamber into their lungs. This is breathing out from here to here, breathing in, and so forth. Just follow it bit by bit like that, and it makes a decent amount of sense, to be honest. So it says compare and contrast the mechanism of expiration during the first 60 seconds of the trace with the mechanism of expiration when they were told to breathe out fully. When it says compare and contrast, it means say what's similar and what's different. But we need to figure out what we're doing what versus what. So in the first 60 seconds, if we look here, that's just the chilling tidal volume. So what it's asking you to actually do is tidal volume, or normal standard breathing versus the visor capacity here. And we know it's expiration because we're breathing out into the tube and the volume of the air is going up when they breathe out into the tube. So the examiner report says that a lot of people didn't read the question carefully and then started yapping about the spirometer trace rather than comparing these two things. You need to look at these two things and compare them as the question has asked you. So the similarities are, when you breathe in, do it yourself right now, your rib cage goes big. It goes, big rib cage. So then if you have a big rib cage and it's going upwards and outwards, you're increasing the volume of your chest or your thorax. If the volume of the chest is big and the thorax is big, then the pressure is low. And then if you breathe out, your chest, your rib cage goes in and the volume of your thorax decreases and the pressure's higher. So if you think about it like a box, this is your rib cage and your thorax, these are the air particles in them, right? 1, 2, 3, 4, 5, 6 of them. If you breathe out, you're now squeezing this 3, 4, 5, 6. You see how those 6 particles are now in a smaller volume, so there's a lot more pressure. Things like to move down a pressure gradient in general to places of an outer pressure. So this is your windpipe out, right? So if you've got a big volume, then 1, 2, 3, 4, 5, 6. These will move in down a pressure gradient, right? Pressure inside is lower than outside when you're breathing in. When you're breathing out, you are now squeezing these, so they get squeezed out into the atmosphere like that. It's the same when you're squeezing a water bottle, right? 1, 2, 3, 4, 5. That's a chilling water bottle. If you squeeze it, 1, 2, 3, 4, 5, the pressure's really, really high, so it just shoves out like that down a pressure gradient. So all of this stuff that we're doing when we're talking about ventilation and breathing has to revolve around changes in volume and pressure, as well as what's leading to it. So we're looking at expiration, so we're going to talk about expiration. So the similarities are that in expiration, what you have is you have the diaphragm and your external intercostal muscles relax. And then we need to talk about what happens next. If they relax, then the thorax volume decreases, or the thoracic volume, you can call it that, or chest volume, decreases. The rib cage moves down and inwards. And then what happens is you decrease its volume, you increase the pressure, and then the pressure in the thorax is greater than the atmosphere. So air moves out of the lungs to the atmosphere, up the, trachea and so forth, down a pressure gradient. Both of them do that. We need to look at the differences between the tidal volume and the visor capacity in this context, normal breathing versus forced expiration. So the difference here is you do it yourself. When you breathe in, you breathe out. It's not, the breathing in is the active part. When you're breathing out, everything is just relaxing. It's passive. Otherwise, if you had to breathe in forcefully and breathe out forcefully, you would be exhausted. When you're going, take a deep breath in and then force as much as you possibly can out, you feel a bit weary, or a bit like, oh. You feel like a bit like, ooh. That's because that forced breathing out, forced expiration, is an active process. So there's something different happening this time. So when you are forced expiring, so forced expiration, what's happening is that this is an active process and it requires energy. Somewhere along the line, muscles have to be contracting because all muscles contract and relax. That's all they can do. So something is making you shove air out harder, which means something has to be contracting. So what's happening here is the difference is your internal intercostal muscles. These are the only times that they're doing their job. Intercostal muscles contract. Therefore, this pulls ribs down further, and this decreases the volume of the thorax and the pressure increases of, the thorax more. And as well as that, when you're doing this, when you breathe out really hard, you can also feel your tummy contract as well almost. What also happens is sometimes your abdominal muscles contract. We need to describe how that leads to these changes in the volume and pressure. Remember, so we talked about the ribs being pulled down and all that jazz. When you're contracting your abdominal muscles, you are forcing the diaphragm up, and that decreases the pressure further. And that's the difference. The only part that's different about forced is that you've got the internal intercostals and those abdominal muscles. So again, with all of these things, you can see I'm talking about the muscle stuff, then what they're doing to the rib cage / abdomen where relevant. Then I'm talking about the pressure and the volume of the thorax, and or wherever. And then I'm talking about the movement of air down a pressure gradient. You need to talk about everything step by step, otherwise you do not get the marks. When it says compare and contrast, you're talking about the similarities and contrast as their differences. And it's asking about the mechanism of expiration, so how we breathe. You do not know if you're talking about anything to do with the spirometer as in describing it. So be careful to just read the question and answer what the question is asking you to do. Complete the following statements about exchange services. So large organisms have a, large something. So they have the options here. They have a large surface area, because we do. If you look at me as a person, I've got lots of places for me for the surface area, but the difference is I have a small surface area to volume ratio. If you flattened a human being like this, right? Or let's turn them into a cube. Let's pretend a human being is a cube. If you flatten a cube, it's got a pretty fatty surface area here, and on the other side as well, but its volume is pretty small because you can see there's not much, thickness to it. If you take that the same cube and then you make them like this, the volume is huge. The surface area is not proportionally as increased. So when you're generally, there's a law for this. It's called the square. This is a bit beyond A level, but it's called the square cube law. And what that says is that when a shape grows, its volume grows faster than its surface area, and and so forth. So the real life example of this, just real quick, is when, for example, if I have 10 centimeters and I want to of a length, and I want to change the 10 centimeters to 100 centimeters, right? I'm timesing by 10. But then if I do that, let's say it's a square, right? Or a cube or whatever. Let's just pretend it's something with a 10 by 10 that gives you the surface area. So its surface area is 10 centimeters cubed. I'm timesing it by 10, right? So this, because it's squared, now becomes times 100. So its surface area now becomes 100 with 2 more zeros squared. But then if I do 1,000 centimeters cubed with the volume, if I times that by 10, I'm actually timesing this by 1,000. So then I end up being 1 million centimeters cubed. So its volume goes really high, really fast. Another funny story is I was playing Minecraft with one of my friends, and I'm not a big Minecraft gamer, by the way, but there's this thing called World Edit where you can make like spheres and objects and stuff, right? So he made a sphere that was about like, what, 1, 2, 3, like times 10 blocks wide, right? He did this exact thing. So what he did is he had a 10 block wide thing. So if it was 10 blocks wide, its volume would have been whatever, 4/3 pi r cubed, whatever, blah, 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 right? It would have been something, so that 10 blocks wide, it would have been something squared and then something cubed. This guy tried multiplying its length by 1,000, which, remember, if you times its length by 1,000, its area has gone up by 1,000 squared and its volume has gone up by 1,000 cubed. So this thing's volume had gone up by 1 times 10 to the 9, as in 1 billion times higher. I don't remember the exact number, but it ended up trying to spawn like 20, 30 billion blocks in 1 second, and my PC crashed and it has never been the same since. So real life application of that utterly butchered my PC. My CPU usage was entirely rinsed. So just remember that for your future reference. Anyway, back to this. That was a bit yapping, sorry. They also need so this means they need a specialized exchange surface, and they need, well, what options do we have? We need a circulatory system, because we need to actually, get things around. So we bring things, oxygen in, CO2 out of the lungs, we need to get it around the body. 2 features of an efficient exchange surface system are, exchange surface must be, well, good blood supply to maintain the concentration gradient, and the other one is a short diffusion pathway. Whenever you're talking about anything to do with exchange of exchanging substances, you need to think about surface area to volume ratio. You need to think about the diffusion distance. You need to think about the concentration gradient and how it's maintained. And as well as that, where possible, temperature. Although remember that you can't change the temperature of yourself to go really high, you could die. The concentration gradient maintained bit is what people don't realize. And this is just a side note. If you have a really high to low concentration, the gradient's really steep. However, as diffusion is happening, or exchange or whatever you want to call it, the gradient's getting less and less steep. So it starts off quite steep and then it becomes less and 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 less steep. Eventually, you're hitting the point where the gradient's like very, very shallow, right? So as the concentration, like as you're doing diffusion, it's not like it's just high to low, happy days. It's like as it's happening, it's getting less and less steep. So it's actually getting slower. So the reason we maintain one is to stop that effect happening. So it's always high to low. Otherwise, as things balance out, it will get slower and slower and slower until eventually you're having barely anything moving from high to low because the gradient's not steep. So usually a lot of things generally either they maintain it by, for example, moving it away. So like O2 moving into your blood in your lungs moves away to somewhere else and brings in new fresh CO2 rich blood and low O2 blood, or it gets used straight away. So for example, in your xylem and phloem, when you get to the sink, the sucrose that's taken is immediately metabolized to glucose and turned into starch and all kinds of other things, right? That is so that the gradient stays steep. So that's something that I see a lot of people overlook. Anyway, I hope that helps. Thank you very much. Please like, share, and subscribe. You know the drill.